So good afternoon, everyone. Welcome to our Fall Dean's Distinguished Lecture. I am first very excited, as you can see from this slide up there. I think this is our first Dean's Distinguished Lecture under our new school name. We are now the Charlie Dunlop School of Biological Sciences, thanks to the $50 million donation that Charlie made to us this year. So that's very exciting. And also I have to say, just as a quick aside, as many of you know, I'm also the director of the Alzheimer's Disease Research Center, in addition to being the Dr. Lionel and Faye Eng Dean of Biological Sciences. And just before I got up here, about an hour or two ago, I found out that our Alzheimer's Center just got a score of 1.7, which I guess is the second highest score in the ADRCs. And so that should almost guarantee us to be refunded. So you could clap for that too. So as you know, we've been doing these Dean's Distinguished Lectures for quite some time, and I think this is a great opportunity for us to invite the community to learn about some of the great science that's being conducted here at UCI that really falls under our umbrella of our school's major theme, which is mind, body, world. And that is that for most of us to have a healthy mind, we need to have a healthy body. To have a healthy body, we really need to have a healthy world, because life is truly interconnected. And so obviously today's theme falls under the mind subcategory. And it's a real pleasure for me to introduce our distinguished speaker, Dr. La Spada, who I can tell you UCI has wanted to recruit here for quite some time. And a few years ago, we succeeded in getting him here. And actually, Al and I share a number of things. Okay, number one, we both work on neurodegenerative disorders. Number two, we're both Italian. And number three, we both grew up in a very unique part of Philadelphia called South Philadelphia, which was essentially the Little Italy part of Philadelphia. So, you know, for two South Philly boys, we did pretty good. So it's going to take me a little bit of time to introduce Al because he's so distinguished. First, let me say he's a distinguished professor, as befits him, of neurology, biological chemistry, and also in the School of Biological Sciences in the Department of Neurobiology and Behavior. He also serves as the Associate Dean for Research Development and the Director of the Institute for Neurotherapeutics. He's also the Vice Chair of Neurology and Pathology and Laboratory Medicine. And so I think another thing we have in common is we don't know how to say no. And so I really admire all the work that he does. Our speaker has been just a pioneer in the field of neurodegenerative disorders. The first individual to discover trinucleotide repeat disorders. That was a huge discovery in the field of medicine and life sciences, because it opened up the doors to other trinucleotide repeat disorders. So he has worked on the molecular mechanisms of spinal and bulbar muscular atrophy, spinal cerebellar ataxia, works on Huntington's disease, and ALS. So he's been absolutely instrumental in helping to uncover the molecular mechanisms and to understand the pathogenesis of this. And I think I'll talk a little bit about how his research is even paving the way for new therapeutic approaches. As you can imagine, he has received a lot of accolades and awards, and I apologize in advance. It's going to take me a while to read some of them, but I think they're worth conveying to you all. He was a Paul Beeson Physician Faculty Scholar in Aging Research. He was inducted into the American Society for Clinical Investigation, bestowed with the Lieberman Award by the Hereditary Disease Foundation for his excellence in Huntington's disease research, received the Molecular Mechanisms of Neurodegeneration Distinguished Research Award in Milan, Italy, was inducted into the Association of American Physicians, was selected as a Harrington Scholar for his translational research accomplishments. In 2018, he became a funded member of the Chan Zuckerberg Initiative's Neurodegeneration Challenge Network, and in 2019, Dr. La Spada was selected to present the Sayer Lecture at the National Eye Institute. I think we are all very fortunate to not only have him here as a speaker, but to also have him on our faculty. So with that, let's give a warm welcome to Professor La Spada.
Hey, wow. So that was really a wonderful, warm and extensive introduction. I really appreciate that, Frank. And I've known Frank for years, and it's been a pleasure getting to know you and now having you as a faculty colleague whose relationship I really cherish. It's really an honor to be selected to give this lecture. I was very pleasantly surprised when Frank asked me, and thank you all for coming today. So what I'm going to try and do in the next, 45 to 50 minutes is basically give you an overview of my research, and I'm going to tell you 3 vignettes, and I'm going to end with a shameless plug for the UCI Center for Neurotherapeutics. Okay, so first, I started out really with an interest in inherited neurodegenerative diseases, and 2 of the vignettes are going to come from this side of the slide. And I've had an interest in spinal and bulbar muscular atrophy, Huntington's disease, and spinocerebellar ataxia type 7. The work in SBMA had led me to become interested in ALS4 through my partnership with Craig Bennett, who's an adjunct associate professor in the lab, and now we've become interested in some other ataxias as well. However, as my research progressed over the years, there were a number of pathways that I became interested in, and that led me to really study more common sporadic neurodegenerative disorders as well. So we have a program on ALS that's actually quite extensive, focused on altered RNA processing. We're looking at the role of non-neural cells in Alzheimer's disease and Parkinson's disease, and I'm very interested in the basic biology of aging, so we have some work going on studying mTORC1 signaling. So as I said, 2 vignettes from the left and 1 vignette from the right today. Okay, so how does this story begin? Like any great story, it begins a long, long time ago at a medical center far, far away, and by that I mean University of Pennsylvania, where I was fortunate to be an MSTP trainee and get MD, PhD degrees. And I was, the most fortunate thing was I was trained by this wonderful mentor, Kurt Fischbeck, who was so enthusiastic and said, well, look, I have 3 projects, pick which one you want. And I said, well, I'll work on this really rare disease that no one's interested in because then there'll be no competition, was the logic for it. And I also thought it was a very compelling, interesting disorder. It's called X-linked spinal and bulbar muscular atrophy, also known as Kennedy's disease for the physician who described it in a paper in 1968. Oh, hold on, there we go. Let me give you the title of it. Okay, so this disorder is a lower motor neuron disease. It only affects males. Patients suffer from proximal muscle weakness. They have bulbar weakness. Bulbar refers to the muscles of speech and swallowing. There's also a muscle atrophy and fasciculations, which are like fine tremors. There's no upper motor neuron finding, so it's not ALS, and it's a slowly progressive degenerative disorder. And an interesting feature of this disorder is that men have signs of androgen insensitivity, including gynecomastia, which I think you can appreciate in this patient. And the androgen insensitivity signs and the X inheritance led us to look on the X chromosome and the critical region mapped to the androgen receptor. And so I was studying the androgen receptor and a repeat, thinking I could use the repeat to track the disease. And this is actually the result that led to the finding, which is that I amplified a fragment of DNA that contained a CAG repeat, and the fragment migrated at a higher position in the affected individuals. Carriers had a lower band and a higher band, and normals just had the lower band. And it turned out that that was a CAG repeat that was explaining that difference in the size of the band. And it turns out this CAG repeat is in the coding region of the first exon of the androgen receptor gene. And what I discovered is that every patient with SBMA had an expansion that was 37 or more repeats. So the way these diseases work is we all have repeats at many different loci, and they're polymorphic. But there's a bimodal distribution. If we don't have disease, then we have repeats that fall within this normal range. It's actually 5 to 35 for the androgen receptor. But if you have 37 or more repeats and you're a man, you will get this disease. It's an absolute association. There's no other mutation that can cause this disorder in the human thus far. Anything's possible, but as of today, with over 3,000 patients followed, this is the only way you get it. So this discovery sort of occurred around the same time that another trinucleotide repeat expansion was discovered, and this was back in 1991. And of course, you make these discoveries, you don't know how important this is going to be. It turns out that this is a very common thing. There are now over 60, actually 70 distinct repeat expansion diseases. And it's really amazing. Some of these disorders are rather important. Fragile X syndrome is the most common inherited form of mental disability. Myotonic dystrophy is the most common form of inherited muscular dystrophy. This G4C2 repeat explains the most common form of inherited familial ALS. And what's really fascinating about this, and we can almost do an entire lecture on this today, but we won't, is that there are no other species that have repeat disease mutations at these loci. So these repeat expansion mutations are a uniquely human phenomenon. It's only our species that gets this type of disease mutation for these loci. Okay, so, and these diseases fall into 4 categories. So the category that I made the discovery in falls into what's called the CAG polyglutamine repeat disease category, and there's 10 disorders in that category. SBMA, Huntington's, dentatorubral pallidoluysian atrophy, and now there are 7 forms of spinocerebellar ataxia. And so they share a number of features. They all affect the nervous system, though you can have some symptoms outside the nervous system. They're all dominant. SBMA is X-linked, but it's sex-limited. Only men are affected. They show comparable median ages of onset, usually young adulthood. However, some can be juvenile onset. They're all slowly progressive, but some you have onset as children. There is another very interesting feature of these diseases, and this is a phenomenon anticipation. It actually was described at the beginning of the 20th century for myotonic dystrophy. The phenomenon, obviously no one knew what the genetic cause was. The phenomenon is that the disease gets worse as it's transmitted from parent to child. Worse means earlier age of onset and more rapid progression. The explanation is that these disease repeats, as they're transmitted, they expand in size. And the longer the disease repeat, the worse the disease. And all these disease repeats that are CAG fall in frame to encode the amino acid glutamine, so they're all caused by polyglutamine tract expansions in the proteins. It's the only way you get all of these diseases except for SCA6, which is a notable exception. It's a bit of an oddball for reasons that I won't go into now. Okay, so the first story that I'll tell you about after I explain this a bit more, I think I want to give a little background in terms of you understanding why CAG repeats are a problem. So if you have too many CAGs in the DNA and too many CAGs in the RNA, you end up with a protein that has too many glutamines. So you may ask, well, why is that a problem? Well, the problem is when you have too many glutamines, the protein misfolds. In fact, what we know is that once you reach this length threshold in the mid-30s, you adopt an abnormal conformation. we as a field have been able to make antibodies that specifically detect that conformation, and they'll cross-react with all the different disease proteins that are in this category. So, I think what's really cool about rare disease is when you work on rare disease, you always wonder, is it going to be important for informing like more common diseases? And in the case of the polyglutamine diseases, where we figured out a number of disease mutations in the early to mid-90s, it turned out to be really important. Because that work coalesced with work that was going on in Parkinson's disease, where mutations were found in alpha-synuclein, and it was found that alpha-synuclein is the principal component of Lewy bodies. So in the mid-90s, there was a paradigm-shifting realization, and that is pretty much all neurodegenerative diseases involve proteins that misfold, whether they're like super rare diseases, like the prion diseases, or super common diseases like Alzheimer's or Parkinson's. What's going on is you have a protein that misfolds, it accumulates in the nervous system, it doesn't get degraded, and you get a disease. So you see that across all these diseases. That's really important because it tells us there's a problem with managing protein quality control. So if we can come up with better ways to improve protein quality control in the central nervous system, we could have a treatment for all these diseases. But there's also complexity with these disorders. And the complexity stems from this question that still is a driving question for our field, and that is, what is the basis of selective vulnerability? So by selective vulnerability, what I mean is in these different disorders, you have different parts of the brain or the spinal cord that are degenerating. So for example, in ALS, you have motor neurons in the spinal cord and in the brain, but in other disorders like Huntington's disease, you have the striatum, and in the spinocerebellar ataxias, you have the cerebellum. All of these disorders are strictly inherited or can be inherited. And in the inherited forms, you have a gene that's mutated, and that encodes a mutant protein. And that mutant protein is seen throughout the central nervous system. Indeed, in the case of ALS1, that is caused by mutations in superoxide dismutase 1, those patients, every cell of their body expresses the mutant misfolded protein, but only motor neurons in the spinal cord and in the brain degenerate. We don't know why that is. So figuring out the answers to selective vulnerability for the different disorders would allow us to come up with rational therapies to treat these diseases, and that's why it's an important focus of research in our field. Okay, so I'm going to transition my stories by showing pictures of my lab over the many years. So the first one was the first La Spada lab barbecue in Seattle in 2000. It was the summer, but yet my lab manager is wearing a heavy sweater, so you can conclude what you may. I don't think it's that cold in Seattle in the summer anymore because of climate change, unfortunately. But anyway, so the first story will be about SBMA. And just to explain the disease pathogenesis, what we figured out from a molecular level, I'm going to tell you about our research to figure out the cellular basis of this disorder. So it turns out that the mutant androgen receptor protein needs to get into the nucleus. This is absolutely necessary. It's not sufficient, but it's necessary for disease. In fact, this explains why it's a sex-limited disorder, because women do not have high levels of circulating testosterone, and the translocation into nucleus is ligand-dependent for the androgen receptor. Also, there's post-translational modifications to the AR. That's important for the disease. There's all sorts of things that have been described, acetylation of leucines and ubiquitination and phosphorylation. We won't go into that. And the main thing, though, is we believe it's a problem with transcription. So the androgen receptor is a transcription factor, and the mutant androgen receptor in the nucleus, what it does is it leads to a sequestration of coactivators, and also there are certain targets that basically don't get expressed. VEGF was one we described. Okay, so but now back to the cellular basis of the disorder. And this is a motor neuron disease, and neurologists have always said, oh, it's the motor neurons, that's where the action is. But it turns out that skeletal muscle, we thought might be important for this disorder. Why is that? Well, patients have elevated levels of creatine kinase, somewhere around 500 to 1,000. Normal is usually up to like 80. That's not massively high, like in the inflammatory myositis, but it's not normal. So that was of note. The other thing is when you do a muscle biopsy, you see evidence of neurogenic atrophy. You would expect that when you have motor neuron degeneration, but you also see myopathy. You see that the muscle cells are sick themselves in their own right. And then there's been a number of studies that were done suggesting there was a knock-in model, suggesting there might be some myopathy. If you massively overexpress normal AR, you get neuromuscular disease in mice. And if you overexpress this IGF-1 trophic factor just in muscle, you can ameliorate disease phenotypes in a mouse model. So there were all these ideas, but there was a lack of clarity as to what was going on. So we said, let's do an experiment where we can answer this question definitively and use the power of the mouse to do that. So what we did is we generated a bacterial artificial chromosome that would contain the entire human androgen receptor gene. Bryce Sofer, who was the second person I hired and really incredible for my research program development, was able to fuse 2 BACs to make a single large BAC that would contain the entire very large androgen receptor gene. It's like 180 kilobases, and it contained all the coding exons, and there was DNA 5' to the first exon and 3' to the last exon, which is important because that means the regulatory elements that control where it should be expressed and when it should be expressed in an organism are there. And we also put in this very large repeat expansion, and we put in these LOX-P sites because this would allow us to take advantage of Cre recombinase to remove this first exon and terminate expression of the mutant protein in certain tissues if we wanted to do that. And that's what we wanted to do. And so this project was led by my former postdoc, Connie Cortez, who is a faculty member at USC. So we generated these mice, and they developed phenotypes that were very similar to what you'd expect in the human. The mice were weak. They had problems in terms of their walking. You saw aggregates in the muscle, and the neurons degenerated. They didn't die, but they became smaller. And also, the mice died. They usually didn't live more than about 6 months of age. And so the experiment that we concocted was to take these mice and cross them with what's called a driver line, where you have human skeletal actin Cre recombinase. So Cre recombinase is only expressed in skeletal muscle. And so you do this cross, you get mice of 4 different flavors, and we're interested in comparing the SBMA mice that don't have Cre to those that do have Cre to see what's going to happen. And we want there to be excision of the expression, a termination of expression in muscle. And so you can see in the biogenic mice, there's no more mutant androgen receptor in muscle. But importantly, it's still there in the spinal cord. So what happens to these mice? Well, it turns out they're basically all better. They no longer have disease. As you can see, they don't have weight loss. They do really well in terms of their strength. You can see how their walking pattern looks like close to normal. And they live, well, it turns out they live well beyond these mice. Around 18 months of age, they start to have other problems of a different nature, but they certainly don't die at 6 months of age anymore. So what this tells us is that motor neurons are dying in this disease, but what's driving the process, we think, is something going wrong in skeletal muscle. So expression of the mutant androgen receptor protein drives the death of motor neurons in SBMA. Well, how does that happen? Well, there's 2 possible explanations, and they're not mutually exclusive. One is that SBMA skeletal muscle no longer produces a trophic factor like IGF, I noted earlier, required for motor neuron health. The other possibility is that the SBMA skeletal muscle generates a toxic factor that crosses the motor end plate, goes from skeletal muscle into motor neurons, and sickens those motor neurons, sort of a gain of function toxicity. So we're still trying to figure this out, and we think, of course, focusing on the neuromuscular junction is really important and very interesting, and this work is now being led by my postdoc, Anastasia Gromova. Okay, so that's the SBMA story with the cellular basis being due to skeletal muscle is what we believe. Okay, so the lab grew a little bit, as you can see. So in the next vignette, I'm going to tell you about another CAG polyglutamine disease, and this will be spinocerebellar ataxia type 7, or SCA7 for short. It's an autosomal dominant cerebellar ataxia that can be readily recognized clinically because patients also show retinal degeneration. There's about 1,000 affected individuals in actually in the United States and Canada. There's probably another 500 in Mexico. And one prominent and tragic feature of this disease is pronounced anticipation. I follow many patients who are teenagers and young adults, and I've cared for individuals who are 2 or 3 years old who died from this disease, tragically. The gene was mapped to chromosome 3 in 1995, and it was cloned in 1997 as a novel gene called ataxin 7. And I became interested in this disorder when I was a postdoctoral fellow at the University of Washington and started working on it then. It's an 892 amino acid protein, and when it was identified, its function was unknown. What was known is that it could go into the nucleus and come out of the nucleus because it had a signal that would take it into the nucleus and allow it to transport itself out. And the range here is 7 to 34 for normal individuals, and 37 up to 400 for individuals affected by this disease. Now, as a postdoc, I chose to work on this disease for 2 reasons. One is the anticipation. I found I was very interested in repeat instability and did a lot of work on that topic. But the other thing, of course, is this question of selective vulnerability. And because this is the only disorder that has retinal degeneration in this polyglutamine disease family, I thought maybe that would be a hook to figuring out, selective vulnerability. So there is photoreceptor degeneration in SCA7. Patients complain of decreased central acuity and a loss of color vision. The first thing is they can't tell the difference between yellow and blue. If you do electroretinograms, these are electrophysiological tests to study like retinal function, you see that the cone photoreceptors that regulate color vision, that they degenerate first because they'll be abnormal and the rods will still be normal initially. But ultimately the rods then go and patients have both poor central vision and peripheral vision, and they are legally blind. And this pattern of pathophysiology allows SCA7 to be described ophthalmologically categorized as a cone rod dystrophy, not a macular degeneration. And there's only, at the time, there was only like 7 genes. Now there's like 10 that are known to cause this phenotype in the human. So my approach here again was to rely on the wonderful mouse as the model. And what we did is we used a standard transgenic approach. We used David Borchart's murine prion protein expression system, and we made a construct with a normal length CAG repeat and one with an expanded CAG repeat, and then we made our mice. And lo and behold, when we looked at the retina, we saw that there was a death of photoreceptors. You can see this scalloping. This is a Richardson stain. You didn't see this in the mice that expressed a normal length glutamine ataxin 7. And it turns out we could do the histology and we could determine that these were primarily cone photoreceptors that were dying. However, perhaps more importantly, if we did physiological testing, if we did electroretinograms, we could see what the visual function of the mice was, and the mice do go blind. However, what I want to emphasize is there's a degradation of cone responses that precedes the degradation of rod responses. Cones die before rods. So this actually was a really quite perfect recapitulation of what you see in the human, this phenotype. So that was very encouraging. And when we looked at sections of the retina and we stained for ataxin 7 protein, we saw these aggregates in the nuclei. We saw them in the photoreceptors, we saw them in bipolar neurons in the intermolecular. I should tell you, this is the retina. It's a beautiful structure, and it's at the back of your eye, and light comes in this way, and the action of visual signal transduction occurs in the photoreceptor layer that lines the back of your eye. And these are actually neurons that give rise to the optic nerve that go to the occipital cortex. But you could see like aggregated ataxin 7 in the nuclei of these neurons. And so this was great, a really wonderful discovery. I tried to publish it in a journal, and they're like, well, this is all wonderful, but give us a mechanism, So I was like, okay, you want a mechanism? We'll give you a mechanism. So my hypothesis was that the polyglutamine ataxin 7 might be interfering with transcription, and it would be binding with a transcription factor that had a lot of glutamines in it. It turns out that a theme in transcription is there are many transcription factors that have glutamine-rich regions. So the model that I thought of was Well, what happens is that the polyglutamine expanded ataxin 7 goes into the nucleus, and there's a transcription factor that has glutamines that is happy and doing its job, except once that goes into the nucleus, then it's sort of sucked away and it engages in an interaction with this mutant protein, and then it no longer is able to transcribe its target genes. And what I came up with as a logical candidate was the Conrad homeodomain protein, or CRX. It's a 299 amino acid protein with a molecular mass of about 39 kilodaltons, and it has a polyglutamine-rich region. It binds to a 6-nucleotide consensus sequence and the promoter of various photoreceptor expressed genes, and importantly, mutations in this gene in just a single copy cause an autosomal dominant Conrad dystrophy in the human. Now, if both genes are, and so this is an adult onset sort of problem, but if you have 2 mutations and it's also recessive, you'll have a blindness phenotype as a child, so-called Leber's congenital amaurosis. So what we did is we looked to see if there was a physical interaction between ataxin 7 and CRX, and there was. But most importantly, we used a really cool new technology. It was called quantitative real-time RT-PCR, reverse transcriptase PCR, with TACMAN. This was like, the big sort of thing that was happening at the turn of the last century. And we looked at mice that were pre-symptomatic for retinal degeneration. They were just 4 weeks old. And we looked at CRX target genes to see if their expression was down. And sure enough, their expression was significantly down, but non-CRX target genes expression remained normal. And I was fortunate then to convince the editor to send it out, and our paper was published and featured on the cover of Neuron back like 23 years ago, back when people actually read journals, and having something on the cover made more of a difference than it does now, though still people enjoy that sort of thing. Anyway, so to continue with what we did, so then at that point, what became important, I think, to understand selective vulnerability is also understanding what these disease proteins normally do. And so again, I won't go into how we figured this out, but we focused on ataxin 7's normal function, and what we figured out is that it is a transcription factor. This was a very nice collaboration with Bob Rader, who's a very accomplished researcher at Rockefeller. I think he's 81 years old now and still is running his own lab there, by the way, which is very impressive. Or sad, I guess, depending on how you look at it. But anyway, be that as it may, ataxin 7, it turns out to be a core component of this coactivator complex, STAGA. And a coactivator complex is basically what transcription factors rely on to go to the site of where they're going to drive the expression of their target genes, because what they do is they affect the chromatinization. They open up the chromatin, if you will allow me. And the way that this complex does it, it has this histone acetyltransferase activity. And so when you acetylate histones, you can open up the chromatin. It also has histone deubiquitinase activity, and this is important for effective transcription. So anyway, based upon this result and other results, some of which I've shown you, we've come up with a model for SCA polyglutamine toxicity that looks something like this. So normally, ataxin 7 goes into this coactivator complex, and there are many transcription factors that rely on this complex to open up the chromatin and allow them to sort of transcribe their target genes. However, when the protein has a polyglutamine expansion, it still goes into the complex. We showed that biochemically with Bob Rader, actually in stoichiometrically appropriate ratio. But what happens is that the histone acetyltransferase activity becomes impaired and no longer works properly. It becomes inhibited. And so now the transcription factor that relies on this complex to transcribe its target genes can't, because the chromatin has not been prepared in a state that would allow for transcription to occur. And so this is about where we were in 2006. And in the intervening years, we've been studying how transcription dysregulation occurs, and we've done a lot of work to sort of also study other mechanisms of the disease process, and we relied on sort of bulk RNA-seq analysis, and those experiments were very helpful in terms of figuring out the importance of certain signaling pathways involving SIR2 and involving calcium homeostasis. But then, of course, there was this wonderful development in our field, which is the ability to sort single cells or single nuclei and to do sequencing, in that way, which we wanted to do because the question we're interested in is, can we define mechanistic pathways in a cell type specific manner? And this work was led by my former graduate student, Luke Bartelt, who's now a postdoc, and Luke, for some reason, provided the picture from his prom to me. But anyway, be that as it may, what we wanted to focus on really was Purkinje cells. And again, the cerebellum, like the retina, has beautiful anatomical architecture. And there's these sort of layers where what you have is the Purkinje cell in this middle layer. You have all these granular cell neurons in this other inner layer. But in this outer layer, the molecular layer, is where these Purkinje cells have all these beautiful dendrites that are receiving information from other sort of neurons in the cerebellum and even from outside the cerebellum, in particular from the brainstem. This, of course, was what Cajal focused on with all those beautiful prints he did back in the day, the beautiful Purkinje cell neuron. And we focused on it not just because it's beautiful, but also because its dysfunction has been shown to be central to the development of the ataxic phenotypes that occur in SCA7 mice, but more importantly, in SCA7 human patients. Okay, but studying Purkinje cells is not straightforward, because they represent just a tiny minority of cells in the cerebellum. So Evan McCosco published this paper where he sequenced like over 300,000 nuclei from mice, from the cerebellum. And that took a lot of money, but I guess they're in Boston at MGH and Harvard, they have a lot of money. So, but only 2% of the nuclei that were recovered were Purkinje cells. So if we wanted to study Purkinje cells and do this experiment, it would have cost like $500,000. So what we did is we said, okay, well, if granular neurons are so abundant, is there a way we can reduce their representation? And we came up with a chemical method of salt precipitating granular cell neuron nuclei. What it involves is using a potassium sulfate-enriched buffer, and then we can precipitate out the granular cell neurons, which, by the way, are the most abundant neurons in your brain. And this, we've published the method if anyone's interested in Cell Report's methods. So we decided that we would do an experiment where we would use SCA7 mice, 2 males, 2 females. We would choose a time point that was pre-symptomatic, and then a symptomatic time point. We would do this Purkinje cell enrichment protocol, and we would do what's called single nucleus RNA-seq. And this paper, by the way, will be out in 2 weeks. And the good news is that this worked pretty well in terms of recovering Purkinje cell neurons. We basically had greater than 40% of the nuclei were for Purkinje cells, so that's great. So then we were able to sort of determine what the gene expression differences were, and this is a UMAP projection to just, it's a multidimensional way of visualizing differences in gene expression. It would take another lecture to explain it, but suffice it to say, what you see at 5 weeks, there's not much separation. There's only 18 genes that are different between SCA7 and control litter mates. But when you get out to 8.5 weeks, there's many genes, like 300, and I guess that's like 20, that are different. And you can see that with this UMAP projection that the separation is quite clear between the neuronal nuclei that are from wild-type control mice versus SCA7 mice. So when you have this type of result and you have these gene expression differences, what we usually do is we try to see if there's a certain pathway or process that might be important. We use the power of computation. We do what's called a gene ontology analysis. And we did that here, and what stood out as an obvious hit was synapse organization. And this was primarily being driven by genes that were upregulated. So that was interesting. However, there was something else that we became interested in. There was a paper in 2022 that caught our attention about how the cerebellum has this sort of periodic striping in terms of expression. And this story that I'm going to tell you, I think it's so cool, and it shows you how science can be really cool and how serendipity in science occurs and is very interesting. So back in 1989, Richard Hawkes, who's in Canada, he was generating a bunch of antibodies against unknown antigens and basically doing brain sections and staining. His idea was like, well, maybe there'll be certain brain regions where certain proteins are enriched, and that will tell us something about understanding something special about the function of that region. And so he had this one antibody that gave this appearance to the cerebellum in transverse section, where you see this alternating sort of immunoreactivity. It looks like zebra stripes, so he called it zebrin. And then he went to town and basically looked at all the organisms way up and down the evolutionary tree, and it turns out that from bony fish up to us, if you take the cerebellum, you see this sort of phenomenon where you stain against this so-called zebrin protein. It turns out to be aldolase C in the glycolytic pathway, and you see this pattern. However, sharks don't have it. So if you're out there, surfing at dusk and a shark comes and is going to attack you, just remember his cerebellum does not have zebrin striping, but yours does, so you can zig and zag and maybe get away from him, okay? Tell yourself that, even if it's not true. But anyway, be that as it may, this is the phenomenon. And this phenomenon has functional significance. Again, this paper that I'm referencing from the Bonney lab, they used FACS, fluorescent activated cell sorting, to enrich Purkinje cell nuclei, and they did single nucleus RNA-seq. And what they discovered is that in the zebrin stripes, you have genes that are important for metabolic signaling, for cell adhesion, for synapse assembly. But in the non-zebrin positive stripes, which have PLC beta 4, these regions of the cerebellum are important for learning and plasticity and for morphology and connectivity. So then we said, okay, well, so there's a bunch of, and this paper allowed us to sort of figure out which genes are the zebrin cell type genes, if you will, and we decided to look at those genes. We looked at about 360 of them that define the so-called zebrin axis, and we found that many of our 350 genes that are altered, of our genes that are altered, which I guess is 320, turn out to be from the zebrin category. And in fact, if you redo the gene ontology analysis to look for pathway significance, it turns out now that synapse is way down here, and zebrin axis genes as a biological category blows away the rest in terms of the significance. And so then the next really cool part of the story is we contacted Richard Hawkes and asked for the antibody, and he just sent it. We got it the next week. There was no MTA, there was no sort of technology transfer, people getting involved, speaking French Canadian or whatever. He just sent it, and we took it and we stained some mice. And what we saw is, beautiful zebrin striping pattern here in non-transgenic littermates. But if you look at the SCA7 mice, you have an obliteration of zebrin patterning. And that was really, to us, a big, exciting result. So then we wondered, well, okay, this is happening in SCA7. Could this be happening in other SCA polyglutamine diseases? So I have wonderful colleagues, Harry Orr, Stephen Polson, Haley McLaughlin, and we said, can you send us some cerebella? And they're like, sure. So they sent us some cerebella, and we sectioned them, and we did some staining. And we actually came up with an algorithm to quantify sort of the amplitude of the expression across lobule 8. And what we did is we observed a complete, basically loss of the parasagittal striping in the SCA1 mice, and the SCA2 mice, and the SCA3 mice. And we also looked at Huntington's disease, because there's a lot of polyglutamine Huntington that is expressed in the cerebellum. And guess what? HD mice, their cerebellar parasagittal striping is fine. They do not get cerebellar degeneration, but yet they have a ton of polyglutamine Huntington in their cerebellum. Really interesting. So this is something that to us is really a fascinating result. Fortunately, the editor and the reviewers agreed. Now, you may ask, well, this is all interesting, but is there any functional significance? Recall I said that in the upregulated differentially expressed genes from SCA7 mice that there was an enrichment for cell adhesion molecules involved in inhibitory synapse formation. I didn't say the inhibitory synapse formation part. So that drew our attention to inhibitory neurotransmission as something we wanted to study in these mice. And just to remind you the way the circuitry works here, again, the Purkinje cell with its dendrites in the molecular layer is receiving input from many different sort of cell types deep in the cerebellum, from the inferior olive in the brainstem, but there are these inhibitory inputs that are coming from so-called molecular layer interneurons that reside in the molecular layer. And we wanted to see if there was a problem with synapse formation. The prediction would be there would be excessive synapse formation. And sure enough, when we looked for that, that's what we found. We found an increase in the number of inhibitory synapses in the cerebellum, both Purkinje cell synapses as well as synapses between molecular layer interneurons, interestingly. And then, of course, we asked, well, will this have any true functional effects? Will this affect electrophysiology? And so what we did is we generated cerebellar brain slices, and we looked to see what was going on. And here we're monitoring what are called spontaneous inhibitory postsynaptic currents. And what you can see is that there's an increase in the amplitude that's significant. The events don't change. For the excitatory postsynaptic currents, what you see is the amplitudes are the same, but the events are occurring less frequently, and that makes sense because we knew that there was decreased spiking rates, and this was a whole cell voltage clamp electrophysiology experiment that we got some help with from a colleague. So to conclude this part of the talk, I think what we've discovered is that Zephyrin-2 subtype dysregulation is a defining feature of SCA7 cerebellar degeneration, starting at motor symptom onset. This leads to dramatic synaptic reorganization, resulting in increased inhibitory synapse neurotransmission. Breakdown in Zephyrin-2 cell identity is observed in all the other polyglutamine SCAs examined to date, suggesting that transcriptional pathology of this nature is a shared common mechanism of pathogenesis. And the other point that is worth mentioning is that this SCA polyglutamine transcription dysregulation becomes apparent well before symptom onset during development, suggesting this is the developmental process, suggesting that developmental alterations may be an aspect of the disease process. And this has been suggested and shown for Huntington's disease in the polyglutamine field. Okay, so now in the final story that I'll tell you, I think the reason why I chose this last vignette is this is all work that's been done since I arrived at UCI, and there's no way this work would have ever occurred if I hadn't come to UCI. And I should say, I'm really glad to be here. I love it here. I'm having a great time, and I'm just so pleased that you recruited me here because I've just really enjoyed having my career here, and I see no reason why I shouldn't spend the rest of my career here and retire. Maybe like Bob Rader, I'll still have a lab at 81. I don't know, if God allows me to be healthy that long. So anyway, so the final story is going to be about a really devastating disorder, amyotrophic lateral sclerosis, ALS. It's characterized by a rapid and progressive loss of motor neurons in the brain and the spinal cord. Patients become weak, they have muscle spasms, sometimes difficulty swallowing if the bulbar region is involved, difficulty speaking. It's a terrible disease that I'm sure most of you have heard of, if not, hopefully, are not personally familiar with from your family, but it's fatal within 2 to 5 years. Some patients die in as soon as 1.5 years. Some patients live a little bit longer. So it can be caused by inherited gene mutations. That occurs about 10% of the time in familial ALS. But most of ALS is so-called sporadic, 90% of the time. So we believe there's genetic risk factors, and they combine with environmental sort of exposure. So when I came to UC Irvine, I gave a lecture in biological chemistry, and I got to know Wei Li. And he was interested in alternative polyadenylation. And this really struck me because ALS is a disease of altered RNA processing. So what is alternative polyadenylation? So every time you transcribe a gene, you make an RNA, and there's a 5' untranslated region, and there's a 3' untranslated region. So when transcription terminates, basically what's done is that a bunch of adenine nucleotides are added at the end. But the site of where that occurs is dictated by this poly-A sort of machinery based upon information that's in the gene. And for many genes, about more than 70%, you can pick a different site to put your poly-A tail. And the reason why that's important is the site that you will pick will determine the length of the 3' UTR. And there are regulatory elements in the 3' UTR that determine stability of the RNA, the ability of the protein to be translated, and where the message goes. You can imagine in neurons that if your message ends up in an axon and it's supposed to be in the cell soma, that wouldn't be good. So I thought this is really an interesting and important phenomenon that could be relevant to a number of different diseases, including perhaps ALS and FTD. And so what Wei had been doing is he was interested in just single nucleotide changes. We would call these single nucleotide polymorphisms that determine, differences in polyadenylation site selection. So here what I'm showing you is going from a G to an A, you're more likely to choose a poly-A site that's further distal, and so then you get more like 3' UTR read. So you have a lengthened 3' UTR. And he called these variants 3' AQTLs. For alternative quantitative trait loci. And 23% of our genes have variants that will affect APA length in at least one tissue. And so what we did is we formed what I've called the UC Irvine APA and ALS team. It's led by my postdoc, Eric Arnold, in my lab, my former postdoc, Sebastian Mickels, who now is a faculty member in Ohm, and Wei Li's former postdoc, Alan Kui, who now is a faculty member here at UCI. And the reason why we focused on ALS is because Wei and Alan had come across the fact that, well, ataxin 3 is a GWAS, a genome-wide association study hit for ALS. It's a risk factor. So there's variants in this gene that put people at increased risk of getting ALS. And when you do a deep dive on sort of the variants from the GWAS and you try to explain them, it turns out that expression QTLs or splicing QTLs don't. What explains it is variation in the site of polyadenylation based upon 3' AQTLs. You can see it's 97% of the explanation of this genetic association in this gene, okay? So our hypothesis then is that increased usage of the distal poly-A site in ataxin 3 is linked to increased ALS risk. Why is that? Well, when you have the longer 3' UTR, you have more cis-regulatory elements where microRNAs can bind. Those are the things that these guys discovered on the East Coast, where they just got the Nobel Prize for, by the way. And what happens is that will lead to the degradation of the RNA, and you'll have decreased levels of ataxin 3 protein. Why would that be important? What does ataxin 3 do? Turns out ataxin 3 is a deubiquitinase, and it regulates protein quality control. It does that by modulating the length of these ubiquitin modifications that are put on proteins when they are told that they're basically when the cell wants them to be degraded, okay? It interacts with a number of important factors in doing this. So ataxin 3 is involved in turning over proteins, and we talked about how protein turnover could be a problem in the nervous system in neurodegeneration. And so in ALS, what is the problem child in terms of protein turnover? Well, it turns out it's this protein that we now know to be TDP43. This is a main driver of disease. It was back in 2006 where we had a pivotal turning point in the ALS field when it was discovered that TDP43 histopathology is a defining feature of ALS. This was done by Manuela Neumann, working in the lab of Virginia Lee and the late John Trojanowski. And it turns out that virtually all ALS patients, certainly all sporadics, most of the familial ALS patients display hyperphosphorylated TDP43 aggregates in the cytosol of neurons, as shown here. And accumulation of the aggregated TDP43 in the cytosol is accompanied by a clearing of TDP43 out of the nucleus. Now, it turns out TDP43 is an RNA binding protein, and while loss of TDP43 nuclear function we think is a key initiator of disease, the accumulation of C-terminal fragments of phospho-TDP43 in the cytosol may also be toxic. So we wondered if ataxin 3 could be involved in TDP43 turnover, and so we did an experiment where we expressed C-terminal TDP43 in two different cell types, cell lines, and we also, what we did is we knocked down ataxin 3 using what's called an shRNA. We reduced the expression of ataxin 3, and you can see then that the C-terminal TDP-43 fragment accumulates, and the phosphorylation of it also becomes apparent as an increase. However, if you overexpress ataxin-3, you can actually reduce that phenomenon. And we did this in transformed cells. We used this TDP-43 that was linked to mCherry. The result was so striking that before you even ran like the blotch, you could see the accumulation of the cherry color in the cell pellet where you've reduced the expression of ataxin-3. And of course, transformed cell lines are nice to do studies in, but it's more important to study neurons. So here we did the experiment in mouse neurons, and we saw the same phenomenon. If you reduce the expression of ataxin-3, you'll have an accumulation of the C-terminal fragment of TDP-43. And while mouse neurons are better than transformed cell lines, the best model organism is the human. So getting help from Len Petruccelli and Mercedes Prudencio, who are at the Mayo Clinic in Jacksonville. They have many patients who have ALS and ALS FTD, and we asked them to sort of do immunoblots, look at the level of ataxin-3, and they saw, hey, ataxin-3 seems to be down in a number of patients. But what's really nice is they have quantified the burden of phospho-TDP-43 pathology in these patients, very quantitatively. And then you could see if there was a correlation between the level of ataxin-3 and the extent of phospho-TDP-43 pathology, and there is. There's an inverse correlation. The less ataxin-3 you have, the more phospho-TDP-43 pathology you have in the brains of these patients. So at this point, we're very fortunate that we got a grant from the CERM to pursue this work. So now we're going to try and figure out the functional significance of this 3' UTR length phenomenon in stem cells that we're making into motor neurons. We're going to use patients who have the risk alleles in comparisons to those who don't have the risk alleles. And we're also trying to make what are called antisense oligonucleotides to block the use of the distal polyadenylation site, see if we can do this in motor neurons in culture, stem cell-derived motor neurons, with the hope that we could restore use of the proximal polyadenylation site to restore ataxin-3 expression as a potential treatment for this disease. Okay, so you've toughed it out really well here. So I'm just about done. So now I'm going to tell you very briefly about our Center for Neurotherapeutics, because the main reason that I think many of us in this field do what we do is we want to come up with therapies to help patients with this disease. And part of the reason why I came to UC Irvine is there were a number of faculty leaders, Dean Michael Stamos, medicine, in pharmaceutical sciences, and Frank and others, who said this is something that would be important and we can support this. And I think the timing's right, because we've made enormous progress in understanding pathways and mechanisms of neurodegeneration. Now we want to apply novel approaches to come up with therapies, but there are barriers, there are bottlenecks. Many basic researchers are simply not comfortable with going from what we would call targets to therapies, and we need more help from people in the physical sciences. So the goal of our center is to, is to create a transformative program that will enable researchers here at UCI to productively pursue translational neuroscience. So our program has two components. We have a drug development unit that I'll tell you a bit more about. The goal here is to come up with novel approaches to discover chemical matter, small molecules that can become drugs to treat neurological diseases. We have 7 funded projects so far that we are supporting. And we also have a program to seed projects that will allow us to come up with better methods to deliver complex biologicals into the brain. And here we're trying to bring together people in the faculty in the physical sciences and engineering with those of us in the biological sciences, and we've awarded 3 grants to faculty teams so far. But to tell you a little bit about our drug discovery unit, what we're trying to do here is we're trying to basically take validated targets from our collaborator labs, individuals who've applied for support and are selected through a competitive RFA process, develop assays for the protein target of interest that can be deployed as high throughput screens, and then be able to screen for the targets against virtual libraries using in silico, basically doing chemistry using computational approaches, or using what we now are creating, DNA encoded libraries, where you have a massive number of compounds that you can screen using this DNA encoded library approach that we're just now deploying, and also doing medicinal chemistry and doing chemical optimization. And just very briefly, this is our team. We have these wonderful research project scientists who do all the heavy lifting in the lab, and we're helping to direct it. And really what I want to emphasize is I have these wonderful faculty who are helping me, who are doing this as volunteers, taking time because they believe that this is worth doing. And that to me is phenomenal, and I think it's emblematic of the way UCI operates, that I can actually find people who are like, sure, I'll take time out of my schedule to help to make this work, and without them there'd be no center. So it's really incredible. And just very briefly, I'll point out that we have faculty in the biological sciences whose projects are being pursued at our center. Leslie Thompson is interested in PIAS1 as basically it's a simulation enzyme, and we're trying to come up with inhibitors. So we've been doing a bunch of things. I won't go through it in detail, but suffice it to say, a lot of effort is being done to come up with small molecules that might be able to inhibit this protein specifically as a treatment for Huntington's disease. And then we have a rather interesting project from Marcella Wood and Ashley Kaiser. They're interested in ACVR1C, so we're trying to modulate actin receptor activity. They've done a lot of work to show how it's important in long-term memory formation. So we're trying to identify novel agonists. And this work involves basically doing some virtual screening and then doing hit expansion and assay development. The point is, this is complicated stuff, and I'm not expecting you to break it down for you. I just want to show you the sort of things that we think are capable through our center, and we want to sort of spread the word. So if there are faculty who are interested in doing serious translational neuroscience, we want you to apply to our RFAs. Oh, someone's timer went off. That's perfect, because I'm done now. I don't know if that was for me or not, but it was perfect timing. So I'm just going to end by just thanking my incredible lab. I have so many incredible people in my lab, and I've had wonderful collaborators. And I really want to emphasize, look at this long list of people that I'm collaborating with at UC Irvine. It's sort of unprecedented, really. And it's been wonderful. It's a wonderful place to be. Frank didn't pay me to say that. I really believe it. My lab, we do a lot of stuff, like go on hikes and things, which you can do here in this beautiful part of the country. And the final thing is, I'll just thank the families, who've supported our work by participating in it, the funders, and I should add, there's also an anonymous donor who recently made a significant gift to the lab, but wants to remain anonymous. So with that, I'll stop, and if there's time for questions, I'm happy to try and answer them.
Hey, we have time for a few questions, and let me start off by saying, Al, thank you for a wonderful lecture, and more importantly, thank you for all the research that you do. And I guess I'll start off with the first question, and that is, can you maybe comment on the role that AI might play in terms of the Center for Neurotherapeutics and drug selection?
I think, people probably have heard about how there's these algorithms that have been developed for protein folding. So I think we can do better sort of modeling structurally of targets using the power of AI, and we can deploy that for our in silico computational screening. I think there's other places where we can use AI in assays so that high throughput assays can become even more powerful, through automation. I think those are two main areas that immediately jump to mind. There may be other areas I'm just not knowledgeable enough to know what they are.
Wonderful overview, thank you. Sure. So all of these neurodegenerative diseases that you talked about are age-related. Do you have any information on what about aging contributes or causes the onset of these symptoms?
Yeah, no, I have a lot of ideas about that. So I think what's really interesting to think about is, the neurons that basically we get from when we're fetuses to young infants, and that develop these circuits and pathways, they're going to have to serve us for the rest of our lives. Sure, we can make some neurons through stem cells in our brain, but that's a very minor sort of percent, and we're not going to replace all of our circuits. So we have to keep these neurons healthy and happy as postmitotic cells for decades and decades. Well, the way we do that is we have processes of protein quality control and also of energy metabolism, because neurons are really energy hogs. And these pathways and other pathways are important for maintaining what we call homeostasis. But the problem is, as we age, these pathways decline in function. And then you have a tipping point where you no longer can keep up with the decompensation that's occurring. I mean, we know this to be true because if you think about it, in a disorder like Huntington's disease, an individual doesn't become symptomatic usually until they're in their 30s, 40s, or 50s, but yet from the time of when they're an embryo, they're making this huge mutant misfolded protein, but yet their neurons are dealing with that stress for many, many decades. But the problem is, as they're aging, these pathways decline in function. So I'm very interested in trying to understand what regulates these pathways to see if we can boost the function of these pathways to come up with therapies for neurodegenerative diseases. So I think that, at least that's the way I think about how the aging process connects to neurodegeneration as probably a major risk factor, certainly for the common sporadic disorders.
Point's well taken, right? We want to solve aging as well, not just neurodegenerative disorders.
Yeah. Well, I think neurodegeneration is, I have a slide on this, a decompensated form of aging. It's basically aging that is not going the way it should.
Exactly. Okay.
Hello, Val.
Hello.
So I know you know because you're on my committee. So what is your thoughts about the polyglutamine diseases and their polyQ expansions and their potential to be involved in things like biomolecular condensates and changes in phase separation?
Yeah, wow. So this is a really complicated topic that Jermaine has brought up. There's this whole sort of new biophysics that really has become clear over the last, 15 years, where we have this understanding of how proteins interact with one another based upon going out of different phases, a liquid phase to a solid phase, and liquid. It's called liquid-liquid phase separation. Again, it's a lecture unto itself. There's no doubt that proteins that are disordered like this are probably engaging in these types of phenomena. I think there's good evidence for this for a number of diseases. I think for the polyglutamine diseases, it hasn't been studied extensively yet, but I suspect that such studies will be very fruitful when they're pursued.
Yeah, good talk. I'm just curious, and the CAG repeat, because there are too many, is there any way to cut it or reduce it and use the gene therapy or something?
Yeah, so this is something that people have been thinking about and working on. In fact, no less than Jennifer Doudna, the Nobel laureate at UC Berkeley, is interested in trying to solve this problem. The problem, but it's a thorny problem, because repeats are a very difficult substrate to sort of manipulate, by doing genome editing. They're like the hardest possible target that you could work on. So while it's theoretically possible, the reduction to practice thus far in the field has not really seen any evidence of success.
Regarding anticipation, is it still the case that it's worse if there's paternal inheritance?
So it has to do with the locus. So everything is sort of driven by the locus. So for example, let me give you an example. So the androgen receptor gene, if someone has 45 repeats and they transmit it to their child, it doesn't change much. But at the ataxin 7 locus, if you have 45 repeats, it can go up to 300. in androgen receptor, it'll change maybe by 2 or 3. Why is that? It has to do with the genomic context. So we know that's important. And the germline, it depends on the disease. For example, in SCA7, the inheritance through the paternal germline, you see much larger expansions than through the maternal germline. However, in myotonic dystrophy, the only way you can get congenital myotonic dystrophy is to have inheritance through the female germline and have the really large expansion. And we know in Fragile X syndrome, the only way you get a repeat expansion that causes disease is by transmission through the female sort of germline. So it depends on which repeat disease you're talking about.
Okay.
But for the CAG polyglutamine diseases, it tends to be paternal transmission is worse in terms of the anticipation.
Like with Huntington's, yeah.
Yep.
Yeah, Craig, you had a question?
I was just curious about the point that you made earlier about the polyglutamine expansion hitting particular loci, but only in humans. I'm just curious as to why that is the case.
Yeah, so this is, I mean, this is such a fascinating, question. Repeats are only unstable through the human germline. So when we put these large repeats into other organisms, they deal with them pretty well. Even if you put them in rhesus, macaques, like there's a Huntington model, the repeats don't change that much. In mice just put in a huge repeat in our germline, we don't care. So I think the implication is that for whatever reason, whether this happened by chance or there was selection for it, we have a germline that's very loosey-goosey with repeats. And we know that this sort of makes sense because we as a species have a very high rate of spontaneous miscarriage. Other species, our closest primate relatives, don't have that. The percent in like chimpanzees from the Xerces breeding program is like 1 or 2%, but for us it's like 15%. So I think that there's some sort of connection there with how we don't handle repeats in our germline the same way any other organism does. And whether that happened for a reason, you can speculate, and I have speculated that it gives us more flexibility in the different types of RNAs that we can form and go in our nervous system and allow our brains to do things that other organisms can't. But that's just speculation. But the fact is it's human specific, and the repeats are not unstable in the germlines of any other organism to date that we've looked at. So that's the sort of where we are right now with that.
Okay, we'll take one more question and then we should break for the reception.
Frank mentioned AI, and it just occurred to me that maybe you could use AlphaFold AI type analyses to simulate the number of repeats in a given protein. I mean, you have several to select from, and to see whether or not there's for that given protein there's a tipping point where the misfolding occurs, and then with increased repeat, that misfolding becomes even different.
Yeah.
And in other words, they're conformers.
Yeah, no, I think it would be great. I had the pleasure when I was very early on in the field to go to a Gordon conference and get to hang out with Max Perutz, because he became interested in this problem, and he did modeling and came up with this idea of beta pleated sheets, and so he did modeling based upon the tools that were available at the turn of the last century and was very interested in this problem. If he were still alive, he would want to probably do the very experiment you're proposing. I think there's a lot of disorder, so you would have to have AI that would somehow be able to account for that in the modeling process, but I guess as we build more powerful algorithms, it should be possible.
Okay, with that, let me just give you a small gift, Al, just a small token of our appreciation.
Thank you.
And let's thank Dr. La Spada again for a wonderful lecture, and more importantly, for all the work he does. And, I always end these presentations by saying we're kind of like PBS. We always depend on viewers like you. So if you're interested in supporting the series, come up and talk to me or Hazel or Madison, and we would love to connect you. So thank you all, and we'll see you next time. And I think our distinguished lecture will be focused on depression. So see you in a couple months.